working. Got batteries today. So it looks like everyone's ready for the game. Exciting. There's more energy today than yesterday. both from myself as well. I was really tired. I slept so long last night. So I'm also ready for the game. I can learn about Brazilian football. Best way possible. So today we're going to talk about social contagion and the difference between social contagion and disease contagion. And it starts off with actually a really difficult experiment to conduct. It sounds really straightforward. So you think if you're interested in social contagion, all you need to do is scare some individuals, such as the startle response of these fish, and then watch it propagate through the group. And people have done those experiments. But the problem is, if I sort of wave at the fish or have a visual stimulus for the fish, how do I know which individuals saw the stimulus directly versus who are being sort of infected by the behavior socially? Right? It's really difficult because fish, in fact, they have what's called Snell's window, this window outside the water where they have a fisheye view of the whole world. And so people have done a bunch of experiments, but they can't differentiate between the phenomenon. So the famous psychologist Ernst Weber from Weber's law, he said the same thing about when people put up their umbrellas, is it because they're copying other people who are putting up their umbrellas or because it's raining? Right? And there could actually be a combination of both. And so this is why it's so difficult. So we tried all sorts of things like using a laser that would track a fish in real time and then zap it in the eyes. And that caused the startle, and that propagated through the group. And we did that for about a year before my postdoc decided to do the experiments in the dark. And the laser stimulus was very brief pulse, and yet we saw massive response immediately because the laser scattered in the water unbeknown to us. We tried to fix that problem, and then we realized there was a tiny click in the circuit that triggered the laser that they could hear that gave them a global response. So in the end, we had to develop a very thin filament that you can't even see that comes up and tickles the belly of a fish. And that causes it to alarm without any others alarming. But they will also alarm spontaneously if you wait long enough. They live in a very, very dangerous environment. For this particular species at this age, there's around 98% mortality per year due to predation. So only a very small subset of the babies ever make it to adulthood. So it's a very, very important selective pressure. But you can see here now, we've developed a system where we can know who is triggering the response, and then we can see the wave propagating through the group. So we did 205 experiments where we knew exactly who initiated the contagious response. So it's like knowing patient zero, so to speak, in an epidemic. Actually, I've heard it's not patient zero, it's actually patient O. It's been misinterpreted as patient zero. You guys probably know more about epidemiology than me, given that you're doing this. But if you look at on a log-log axis at the number of events, the number of cascades, and the size of these cascades, far too little data to say anything about whether this is a Powell or anything like that. But nonetheless, it certainly has a long tail. So most of the events don't go anywhere, or one or two individuals respond. Some of the events, it percolates through most of the system. So certainly very unpredictable as to whether it's going to lead to a massive outbreak or almost nothing at all. So we were talking yesterday about these very simple models of social contagion. For example, the metric, the topological, the Voronoi, and the very simple visual model. And you saw that even if we assume it's vision and we don't really know much about which visual cues it is, it does better than the simple models. But it doesn't do brilliantly. So what we needed to do was really establish the functional mapping between the sensory inputs, the visual inputs that they're using, and the motor outputs. So as I mentioned the other day, what all brains have evolved to do is to translate high-dimensional sensory information, in this case visual information. So this is just the visual field reconstructed, wrapped around to the left eye, to the right eye, and the color or the lightness represents how far the object is away. If it's black, the object is very close. If it's white, it's the wall of the tank. If it's light gray, it's a fish that's further away. So you can see, even if we look at this one-dimensional, or two-dimensional, because time is on this axis, representation of the time-varying visual field, it's incredibly complicated. Yet the brain has to take that information and turn it into a low-dimensional behavioral output. And alarm response is interesting from very many perspectives. Firstly, predation, as I said, is the main driving selection pressure. Secondly, the alarm response is triggered predominantly by two giant neurons called Meissner cells. So it's a reflex response. So the brain has to translate this down to a first approximation to a very small number of cells that then make this incredibly important decision on the part of the animal. So we were interested in how does the brain do this? How does it translate this information? Now, that would be a lecture in itself, so I'm not going to go through the details of that because it's not important for the final story. But using the visual field reconstruction and the body posture mapping, so we get the body posture of the animal, and then machine learning techniques, we could reverse engineer which visual cues they're actually using. And we also looked at combinations of cues. And all that really matters for today's lecture is it turns out that they use the simplest cues they could possibly use. They're not using derivatives, for example. Like I would have bet my money it's things like lume, the rate of expansion on the retina, things like that. But they're not using that. They're using relatively simple visual cues, one of which is the ranking of individuals in terms of the area subtended on the retina. So it doesn't matter how far away or how close they are. What matters is your ranking in terms of how big you appear. And they're not compensating for the angle of the body of the other individual. Perhaps it takes too long to do that. They're not even, I would have bet that what they would do is use the body depth of the individuals. Because if you imagine, I think the point was made yesterday, if you've got an individual here, a focal individual, and there's a neighbor in this orientation here versus here, this subtends a very small angle, this subtends a very large angle. However, if you imagine from the perspective of the animal, if they measured the body depth, it doesn't matter whether an individual is pointing towards you, away from you, or sideways, the body depth is invariant. For whatever reason, they do not use that. They use the area subtended. Okay, so I'll just switch back. We figured out that, surprisingly to us, they're using very simple visual features. They're using very robust features, probably because they live in environments where the water is scattered and absorbed by particles in the water. So it's and also anyone that's gone swimming, you see this caustic light environment, right? At the bottom of a swimming pool, you see all the light patterns. It's a very complex light environment. So maybe, we don't know, but maybe that's why they use simple, robust features. But nonetheless, despite the fact that features are very simple, the geometry of the problem is not. The orientation of the individuals. And so what we did was so coming back to what Weber was saying about putting up the umbrellas, here we know that we're looking at social contagion. We know that the other individuals are not detecting something environmental. But when the first individual responds to the stimulus, we can be pretty damn sure that this individual is responding because of this individual, right? But what about a third individual? Did it respond because of that individual as well? Or did it respond to the second individual? Or did it integrate the two stimuli? We cannot tell. We can't be sure. We can only be sure of the first responder. So we built our model of how the individuals respond to the visual field only based on the first responder, because then we could be sure. And it didn't matter where that responder was in terms of the visual field. The model was exactly the same. And so then taking that information, from each moment in time, we can make the assumption that other individuals are somehow using the same model, the same visual features, to regulate their contagion. And before, we figured out that it was a fractional contagion. It was the number of individuals that had responded versus hadn't responded to the stimulus. And so we asked whether that actually also explains this contagious behavior. So if we zoom in, so this is a real fish school, remember? If you zoom in and you calculate the visual fields of all the individuals, this is at the moment of initiation, before the wave propagates. You can see already we have a complex weighted directed network because of the geometry. I might be strongly influenced by you, like an individual here might be strong, this one could be strongly influenced by this one because it's subtending a large area, but this one may be much less influenced by this one because it's subtending a small area on the retina. So we end up having this complex weighted directed network. Now, we cheated a little bit because the wave that propagates through the fish school is so fast compared to the rewiring of the network. We just used that network frozen in time at the initiation as our network. Of course, it changes slightly, but we didn't want to kind of cheat. We wanted to predict from the network before the contagion happened whether we could predict how the behavior would spread through this network. And so here is the network reconstructed. So this is a quantitative network. Often in biology, it's like they use proxies, like who is close to whom. And you can see from this example here that who is close to whom is not good enough in terms of the social contagion. And so you can really see this beautiful network where sometimes there are mutual connections, sometimes there are directed connections, and the weights here represent the strength and the colors represent the mutual connectedness. And I'll just show that again because it's the first time we've ever been able to visualize a real social network in these types of systems. Because unlike the brain, there is no physical form in terms of the connectivity that gives rise to collective behavior.
Are those simulations?
No. These are real fish schools. Nothing I'm showing you here is going to be simulations. It's going to be analysis of real data.
Next T-Rex?
No, this is pre-T-Rex. T-Rex would have made this so much easier. And we would have known identities and all sorts of other things. So we can do this in a much more sophisticated way now. We've actually replicated this experiment with real predators using T-Rex, but we haven't analyzed that yet. The student moved back to China.
Is the video, the network topology not changed?
Yeah, so no, here, this is actually, yeah, you've made a good point. So this is just so we can visualize what these types of networks look like. In the actual analysis, we freeze time at the moment of initiation, and we ask, can we predict who will be infected by this behavior, when they'll be infected, and how far will it spread? We didn't want to do that with a time-varying network, because it could kind of be cheating, like we could kind of be getting out what we put in. So we, in the actual analysis, but that's not to say that it would not be incredibly interesting to analyze these networks. And we have done a bit of that. We have a paper that came out last year. For example, this is not a small world network, it's not a scale-free network, it's got many grid-like qualities, and so it's sort of, I think there's a whole wealth of work that could be done on just analyzing these networks, because these have evolved under selection pressure. It's not like Twitter or Facebook that just happen, they don't have an adaptive value. And so I think, again, the data, by the way, are freely available to anyone that would like to do that. And so what we're interested in here is social influence and how it relates to the network structure. Okay, so let's think about what people used to think. I already kind of gave the game away yesterday that this is going to be incorrect, but I think it's useful to think through what people used to think about social contagion. It's intuitive to think that the most socially influential individuals are those with many connections, a sort of hub within a network, so to speak. And in the network literature, many of you will know this is called a high degree. It means you have many connections to you. And also whose neighbors have a low cliquishness, a low clustering coefficient. Well, why is that? Why do we assume this? Well, it's because people think of and model behavioral propagation, behavioral contagion, as a disease, because it's so intuitive that this should be correct. Because, of course, if I have a certain opinion, and I'm trying to convince you, the more of you I speak to, maybe the more I'll convince. However, if I only interact with the group here at the front, and not with the rest of you, not with the riffraff, just with these cool people here, then I may infect them, but if they're just talking to each other, communicating with each other, it's not going to spread, right? So this is entirely true. And in fact, I think it's just so obvious that no one had ever tested, is it actually true, right? And so I think it's always useful to do these kind of biological tests. So we just asked, not modeling it, actually testing it. Is this the case? We can measure these network properties from our initiator, right? And we have initiators all over the groups. They have all sorts of different local network properties. And then we can just ask, is this true? Turns out no. It turns out the most influential individuals, and this is a strong result, this is not a weak result, were those with a low degree and whose neighbors had a high clustering coefficient. In fact, we had to come up with a new measure here because it's a weighted clustering coefficient.
What is the definition of the clustering coefficient?
I can't hear you.
What is the definition of the clustering coefficient?
It is that your neighbors, I had it on this figure here. Oh, sorry, I didn't have it on this figure here. It's that, yeah, your neighbors are highly connected to each other, is a high clustering coefficient. If your neighbors are not connected strongly to each other, but to other individuals in the group, you have a low clustering coefficient. So sometimes it's called cliquishness. How cliquey are your neighbors? How interconnected are they? And how weakly are they connected outside the group? As I said, we had to slightly modify it to account for the weights, but it's, you can find it in the maths in the paper. Okay, so it's individuals that have few connections to neighbors and whose neighbors are highly interconnected with each other that turned out to be the most influential. Exactly the opposite of intuition and exactly the opposite of what's been assumed when modeling social contagion. At least until this paper came out. So why is this? Well, we can think about it again. This is a loose analogy. But if we think about political views, I had a very interesting lunch yesterday where I heard a lot about Brazilian politics. And so people can have political views, right? And I could try to persuade you that Donald Trump, say, was, you know, the best president and is going to make America great and glorious again, right? But I may not convince you. I hope I wouldn't. I may not convince you. And so there's something, you know, and I could spend all day trying to convince you, and it just may not work, right? But if I have COVID, and you haven't been vaccinated, and you haven't had COVID before, the longer I'm in communication with you, the higher is the probability that you'll be infected, right? So there's something fundamentally different about opinions. And what is that? Well, it's that humans do this, turns out fish do this, all animals we've studied do this. You seek reinforcement from multiple, what you perceive as independent sources before you change your opinion. This is, I mean, like, of course, we now use Google. We can have lazy cheats, but we've evolved to utilize multiple sources. So even on Facebook, for example, people do this. And the way I think about it is, when I lived in Princeton, I used to go to New York City and go and hang out with my friends. And one day I went to New York. They were recommending an album. This is pre-Spotify. This is when you actually get to buy the albums. And they were, you know, talking about it, but I wasn't convinced. You know, am I really going to spend the money on that? But then when I got on New Jersey Transit to go back to Princeton, the couple in front of me were talking positively about the same album. My brain, sort of without me knowing, I bought the album because that was decorrelated information, but it was supportive, right? The way humans do it, there are processes like that. On Facebook, for example, if you get multiple messages from the same clique, the same subset, you don't pay so much attention to the second message or the third message. But if it comes from a different clique, so you have family groups perhaps, you have different friendship groups. So whether it comes from within one of those groups, which could be correlated or not, your brain pays attention to, even subconsciously. Now the fish, what they're doing is they're paying attention to where the information comes in different parts of the visual field. If it comes in from the same part of the visual field, it could be correlated. But the other important thing, because you need this reinforcement, if you have cliques that have loops going through them, it means I could influence you, and you then influence you, and then you can then influence this individual, and then you can influence you again, right? You can have this information coming from multiple sources. So I can influence people directly, or fish can influence fish directly and indirectly. And so this increases the probability. And we find that this is called a complex contagion, right? So it differs from a disease, which is a simple contagion. And one of the coolest things about it is it turns out that, as you can see, the local network properties that maximize the spread of behavior are exactly those that minimize the spread of disease, and vice versa. And I don't know whether that's coincidence or whether that's not, because you could imagine in the evolution of sociality, you want to spread information, you want to spread behavior, but you don't want to spread disease. So I don't know whether that's a coincidence or not. And so, as I said, social contagion is a so-called complex contagion. And shortly after this came out, our fish were on the front cover of this book by Damon Santola, where he discovered very similarly in humans, in a wide range of scenarios. His first paper was in Science, looking at how people make medical decisions. But then he's since looked more recently at microtransactions in India and lots of other scenarios. And it turns out, despite the vast differences in social organization, humans also use complex contagion, and it really changes how networks transmit information. So it's not like the spread of a disease. And there's a YouTube video here, I'll share the link with you, where he describes his book and these common features of social contagion. So I find that quite fascinating. And we've since looked at birds, we've looked at other animals, and this seems to be conserved. And it makes sense. Yep.
So I think I understand the argument for why clickishness could be an important mechanism, but the low connectivity, I guess, is accidental.
Thank you. I completely forgot to go into that. You're absolutely right for picking me up on that. So there's a very famous paper by Granovetter called The Power of Weak Ties, where he argues that many, many weak connections are very influential. It turns out he was wrong. It's a really cool paper, but this was, pre, Facebook, Twitter, all these, massive data set analysis. It turns out both in humans and in fish particularly, a few strong connections massively outweigh many weak connections. It doesn't mean weak connections are not important at all. It's not like an and/or thing. But it turns out that these few strong connections are super important. And I do also wonder about that in human societies. I mean, if people are analyzing Twitter, for example, or your followers, or your friends on Facebook, are they really your friends? There's something divorced in these online social networks between, some individuals on those networks may indeed be influential to you and others not. If you actually look at natural human societies, like the Hausa tribes and others, they tend to also have these relatively few strong connections, even within families or between family groups. So I suspect that this is probably not just fish again. But of course, if you're broadcasting, if you're able to have 2.5 million followers on Twitter and you broadcast nonsense information out there, of course you break all these rules. I mean, this is why these social networks are potentially so dangerous, because many of the strategies and the instincts that we have, because they really are non-conscious behaviors, are because we've evolved in societies where there isn't this mass broadcasting. But we'll come to that in the last lecture. Also, I talk a little bit about that. Well, you can read that. I think you were sent the PNAS paper that we wrote recently. Yeah, there's much more about it in there, but I'll go through it I'd love to get your feedback on that actually in the last lecture. I think we're also going to be pretty exhausted by the last lecture, so I think more discussion, less presentation. Okay, I've actually got the video here, but I guess we don't have sound. But I'll send the slides. Okay, so then, if we're correct, and there's this new way of thinking about networks, and we're correct also in trusting our model, which is, of course, a prediction. We don't even know if this is the right model. We tested as many possible visual cues and interactions as are there in the literature. But there's always a possibility that there's something we didn't think of. And that's why we're now using machine learning to try to find non-human created rules. But anyway, that's a different story. But I don't know whether it's possible to remove me from the corner here.
Next.
Oh.
No, that one.
This
that
one?
one? Yes.
Oh, there we go. So the idea is, can this actually now, remember before, when we had those cascade sizes, it was a Paolo-like distribution. So sometimes it would lead to a massive avalanche, sometimes it would lead to nothing, and we had no idea why. We had no ability to predict. Well, now we run new experiments where we trigger individuals, because you don't want to use the model that you built from one set of data to then test it on those data unless you do it very carefully. So we did a whole new set of experiments where we predicted at the onset, at the moment an individual was initiated, we predicted who would be infected, when they would be infected, and also how far it would spread. And so here you can see this long tail type distribution, but now we can predict, it's biology, right? Remarkably precisely what's going to happen before it actually happens. We can predict whether the contagion will lead to an epidemic, so to speak, in a behavioral sense, and change the whole behavior of the group, or not do anything at all. Now, admittedly, this is a simple behavior, a reflex reaction. These are simple vertebrates, fish, but we were still really happy because prior to this, this was completely opaque. We had no ability to understand what was going on. Yep. The behavioral states here are alarm or non-alarm. So fear, like this fright response, the sudden change of behavior, or not. And the reason we did that is because it's a reflex. You know, it's like when you tap your knee. You just have to do it. So the individuals either show it or they don't show it to a first approximation.
Okay, and it's assumed that that happened just for one individual, or?
We always know it happened from one individual because in these experiments, we ensured that it was only triggered by one individual.
Okay.
Now, there's an infinitesimal chance that another individual, just by chance, startled from an independent source, but it's astronomically small for the time scale of these experiments. I should have explained, that video at the beginning was slowed down. I think it said slow down 5 times. So it's a really fast response across the group. And on average, an individual in a group size of 150 would startle once every 3 or 4 minutes. And the experiments were less than a second or something like that. So we have that in the paper as well, that if the probability of any one of these experiments, including multiple individuals, was very, very, very small.
This difference in the size of the cascades, there sometimes you have a very small one, and sometimes it spreads a lot. Do you think it's this model seems to? Would you say that this model seems to show that maybe that's has more to do with the stochastic distribution of the visual fields? Or do you have I don't know, because I don't know the fish. Do you have some individuals that are more important than others naturally? Some kind of leader or not?
Damn it. So to simplify the talk, I took out some slides. I thought I'd put them as hidden, but I seem to have taken them out completely. Oh no. Well, maybe we can come back to that. If we take a break. So yeah, what it really relates to is the position of individuals within the group. So if I just switch to the blackboard, maybe I can try and explain the general principle here. And again, it's kind of obvious. But if you're an individual, if you're an individual near the front or the side of a group, for example, this individual here has got no neighbors on this side, right? So it's got fewer connections. And so if one of these neighbors were to startle, the fraction of neighbors that startles is greater than if it was in the middle of the group, because it's got more neighbors. What does that naturally mean? It means that the edge of the group is more sensitive than the center. So just spontaneously, the edge of the group is like the sensor array. If individuals at or near the edge respond, it's much more likely to transmit than from individuals in the middle of the group. Secondly, related to this, also in the paper, we reconstructed the visual fields of individuals to see what they could see outside the group. Say there's a food patch here, or there's a predator here. As you can imagine, these individuals have got excellent direct information about that. They also have tight social connections that's very good at transmitting information for the reasons they showed. So these edge individuals have a double benefit. They're both more capable of getting direct information and more capable of propagating and receiving social information. Now, again, I've simplified it again because there's a slight network difference between being good at sending information and being good at receiving information, but that's beyond today's talk. But if you look at individuals at the back of the group, they tend to have pretty good social information because they've almost got surround vision. But they actually turn out to have very poor social connectivity because they're kind of more sparse at the end of the group. And so, in fact, what we found was that no one really wants to be at the back. The safest place, everyone used to think the safest place is in the middle. But the safest place by far are the front and the side edges. And so individuals are constantly jostling for those positions, and that's why the group's on the move. You know, they're constantly trying to get into these, and because people had never put information into the theory before, that this was not seen. And then, maybe I'll show it later, but then we did experiments in the wild with sonar imaging, with 2mm precision sonar imaging, and we find that predators always attack the back of the groups. And in fact, the predators line up to slice the group, because if a single predator hits the group just reforms. And the predator, the information flow, even from the back, is so good that the predator can't really get the prey, especially for large groups, because the wave of information transfer that I just described propagates about 10 times faster than the speed of the predator. So the position of the predator is given up by this wave of propagation to individuals really far away. So it really struggles. But if the predators form lines where they slice through the group, then the predation risk, of the risk of being eaten as a function of group size is like this. It's an extraordinarily steep function. So the predators really have to break up the groups to stop the information flow, because the information flow is so damn fast that they can't cope with it, they can't beat it. And this is from the field, this is from in nature. So it's kind of nice to show how the experiments in the lab that made these predictions about spatial positions actually does translate to the positions seen in nature.
Is there some sort of result theorem showing that there is an implied mixture in the system, so that you guarantee that because otherwise, if you have different areas in which the fish will be predated more or less, they will tend to a strategy, as I think evolutionarily, to move to those areas. So
that's exactly why the group's on the move. No one wants to be at the back. Everyone is jostling to get to the front and getting away from the back. So actually, I think that's largely why these groups are always on the move. But there are other predators, there are other risks, right? If you're at the front of the group, you may also encounter other types of predators. But we've had a paper that came out last year where we looked, so we thought, well, the predators we should, I'll try and show you a video of it because it's really cool. These predators were spotted sea trout hunting Gulf Manhattan off Louisiana. And these are pursuit predators. They're moving really fast through the group, the sort of thing you see on like Blue Planet type thing. But there are many other predators, like pike that we studied, that sit and wait. They sit there cryptic, waiting for the prey to come across. And so we also, last year, studied them. And I thought, well, maybe they're providing the predation pressure from the front of the group. So you got threats from both sides. And that could be true. But certainly for the school sizes we looked at, the pike would also wait for the prey to get over them and then strike. So again, individuals in the middle were more at risk. Here, individuals at the back were more at risk. With the pike, individuals at the front middle were more at risk. So I think, and there's a multitude of different predators. To do these experiments takes at least a year or two. It takes about a year to get permission to do them. It takes like at least another year to do the experiments. And so there's almost too many different possibilities. So I certainly think being at the back of the group is a very bad thing against common predators. Being in the middle of the group is a pretty bad thing against common predators, but there could well be predation at the front of the group that we just haven't detected yet. But certainly it seems no one wants to be at the back. Like they're constantly, and you often find the smaller individuals or the weaker individuals at the back of the group. And this is not just in fish schools, you see this in animal herds as well. The predators will often get the prey to run for a bit to try and pick up if anyone's lame or weak or so it's kind of an honest signal, right? If you're stuck at the back of a group for a consistent period of time, it's probably because you're less good at trying to get away. So the predator will focus on such individuals.
Is the studies on how the predator attacks the group, if they are made on laboratory or they are on the field?
Couldn't get the last sentence.
These
last
studies
field.
on how the predator attacks the group, they are made in laboratory or they are on the field?
So this, let me just show you. I think it's worth me finding it. Let me just disconnect for a second. It's a bit stupid of me to not hide the slides. Let's see. I may have them somewhere. Oops. maybe we can take just a few minutes break, like if you need to go to the loo or something, and I'll just figure out these slides, and then we can reconvene. Okay, folks. Hello, everyone. Ready to go again? I've got the slides. Yeah, thanks for asking such. It's fine. Thanks for asking such great questions. It's always really fun to have this. And of course, there's so much stuff that we've done that it excites me that you're asking really the really coolest questions. And so yeah, so we did, as I sort of tried to explain here on the blackboard, but in a slightly more sophisticated way, reconstruct also how well they could see outside the group, which again is such an obvious thing, but had not been done before. And so just to quantify what I said, if you look at the distance from the group boundary, and we look at social influence, like how good you are at obtaining and transmitting social influence, and so we don't really know how they process the external visual fields, so this is just how much space do you see outside the group. You can see actually being at the edge of the group is really much better. And this is in direct contradiction to the standard theory of William Hamilton, geometry for the selfish herd, which is one of my favorite papers of all time. It's just that people in biology often get stuck in the theories of the 1970s and just conduct an experiment to try to test Hamilton's selfish herd hypothesis, conduct another experiment to try to test his selfish herd hypothesis, without thinking, you know what, maybe things have changed since the 1970s and that we need a dynamical theory. Okay, so as soon as you put dynamics and information in, actually the selfish herd prediction disappears, depending on the type of predator. Okay, so it's not a criticism of Hamilton, it's more a criticism of biologists, at least non-quantitative biologists. And then if we look at the front and the back of the group, as I said, at the front of the group, you've got excellent access to personal and social information. At the back of the group, because of the lower density, you actually have a bit better personal information. There's more space around you, but that's also potentially scary because you also have poor access to social information. And so to sort of test some of these ideas, again, I just threw this in just so you know, this is a paper that we did last year. If you're interested in criticality, we've gone into that story about, I told you we didn't have enough data to say whether this is truly critical or not, but what we can do is, and what we did do with Pavel Romanchuk, who used to be a postdoc in my group and is now a professor in Berlin. And this is, you're not meant to pay any attention to this, but just if you're interested, we used like another sort of a disease propagation model, but with complex contagion, parameterized by the data. And then we did things like rescaling the data. What if they were closer together? What if they were further apart? Why are they regulating density in the way they do? Is it actually poised at criticality? Is this really a power law? And we argue that it's probably subcritical.
I still didn't understand what was the y-axis in the graph that you showed before. It says it's standardized units, but what did you measure?
So we measured here the social influence, so normalized social influence, and the external visual field, which is also, I don't remember what the units are, it doesn't matter. Okay, so this is just to highlight, if you're interested in criticality, if you're interested in these types of phenomenon, it's not that we just were happy that we can't get enough data. We actually tried to then build models and sort of see where our system was poised and argue that it's actually subcritical, which is what people also argue in the brain, that you tend to be in a subcritical state. So you can see that for more information there. Again, this is just sort of advertising that we've also done experiments with real predators, with sit and wait predators, and asked, how do the prey flee? How does the predator attack? So taking the perspective of both the predator and the prey. This is where we found that, again, it wasn't the front individuals, but kind of individuals near the front that tended to be most heavily predated. But I think it is kind of cool to show that we also studied in the wild. So you can see here, I think still beautiful data. It's from 2012, I think, but still beautiful data, where you've got real wild predators attacking these very large fish schools. and we used image processing to get the velocity vectors of the prey and the motion of the predators. And we found that if you look at the prey velocity field, so this is from the perspective of a predator, the color represents the density of prey. So you can see the prey kind of flow around the predator and leave a vacuole behind. They begin to reform. And you can see the vectors mean the prey are moving away from the predator. But you see they're all pointing in this direction, which also shows you that the school is moving this way. So they're attacking the back of the group and moving through the group. Again, this is real data. This is not a model. So the predators do attack groups from behind. So perhaps this information hypothesis that we have is actually meaningful. And how did the predators cope with this? Well, we weren't initially having this study as a collective behavior of predator study. We were thinking about the prey, but we found that if you look at the distribution of where other predators are, if this is a focal predator, other predators tend to be in front of it and behind it. And in fact, at the range at which the prey school reforms. So you can get up to 5 or even 8 in one occasion predators in a line, slicing through these prey schools. And we couldn't actually tell if the predator had eaten a prey. The resolution wasn't good enough. But we could easily tell how close a prey is to the mouth of a predator. If it disappears, it could have gone out of the plane of the imaging, or it could have gone into the mouth of the predator. We can't differentiate, but we can reasonably assume if you're very close to the mouth of a predator, you're more at risk. And if we use that as a proxy of risk, you can see this result I showed you, where the predators really struggle to get prey close to their mouths, unless they manage to slice up the groups into very small groups. We also looked at other indicators of criticality in this paper. We showed scale-free correlations in velocity and speed and so on within the groups, indicative that really you have this very fast information transfer. We also showed the waves of information flow were extremely fast. And so this was all done in the wild, which is kind of makes me wonder why we don't do more of that kind of work, because the technology is even better now. But any questions? No. So that's just kind of an aside to go into some of the questions that you were asking. But I think there's still tons more to be done, especially from the predator's perspective. We've hardly ever considered why is the predator confused when it faces multiple prey? How does the predator adopt different strategies itself? So just to sort of summarize what we've learned so far looking at these fish schools, is that computation can be an emergent property of the network structure as well. Oh, there's another paper I forgot to mention. We published a paper in PNAS last year where we showed that when the world becomes more risky, the individuals change the signal-to-noise ratio of the group. So the group becomes more twitchy and sensitive. But it's not the individuals changing what's going on in their brain. The algorithm they use is identical. They change the network structure, and the network structure changes the information processing. So I think we cite this one here if you're interested. So we've shown that they change the network structure to regulate social contagion, to modulate, actually, it was from more than a few years ago, to increase risk, sensing long-range environmental gradients. Remember that emergent sensing story. We've also shown that these properties readily evolve among selfish individuals. So even though the levels of selection are wildly different, nonetheless, the fact that they're changing computation by changing the structure and the weight of the network of communication is very reminiscent of the brain and how the brain computes. Okay, so I think even though these are selfish individuals, I think that there's a great reason for neurobiologists and those in collective behavior to actually think more and work more together. Because natural selection doesn't care. It's going to find the easiest tricks that it can find for computation. And I think we're focused way too often on the individual brain and not enough on the connectivity between brains. But what I hope to go on to show for the rest of today is how making these kind of connections, I'm going to continue talking about animal groups, but eventually I'm going to turn to actually what it's taught us about the brain and what we're studying now in terms of how the brain functions. But I want to set the seeds now that this is not just about animal groups. This is about spatiotemporal computation, generally speaking. Okay, so I don't know why it's cutting off the slides, but I guess I can share. Pardon me?
Yeah.
I think it's. I think I could try to fix it.
It doesn't matter really. I don't really care. I can share the slides anyway.
Before we move on, I'd like to ask, all of those analyses are done in 2D, and you argued yesterday that you could pretty roughly translate to 2D in fish behavior. Does this hold true for like starlings and whatnot in like 3-dimensional space?
It's really hard to get the data. It's hard to get the data in reality, and also the Italian team that works on starlings refuses to share their data. They're kind of notorious for that. They're great scientists, but I think the data are so hard to come by that they're reluctant to share it. I would say our data are often hard to come by, but we're very happy to share it, so it's just a different philosophy. But I think, yeah, I think this, I think some of the starling results may be things like gusts of wind and so on. There's been beautiful work from Stanford, Nicolette, who's shown that the starling people also worked on midges and showed these scale-free correlations in midge groups. There's no reason to think that midges that are trying to mate with each other and compete for mating should evolve some sort of information processing at the collective level. So I was a bit sort of, that's weird. And then Nick did these experiments in the wild and in the lab, and in the lab this completely disappears. In the wild it's driven by air currents. So it's the same when, that's why I'm cautious about the scale-free correlations we see in the ocean. Is that due to the behavior, or is that due to the ocean currents? And there have been other papers whereby fractal type behavior or levee flight type behavior that's been seen in the ocean can actually now be explained just by the physical structure of the ocean, not by a strategy. And so I think it's really hard to do some of these studies. I'm also quite cautious. I hope that these are general principles. We have studied starlings in the lab, but only in relatively small groups. So starlings are just really hard to study. But you also mentioned 3D. Extremely important point. These studies are so hard to do that it takes us, as I said, a year to get permission and a year or more to do the analysis, many years sometimes, and that's in 2D. In 3D it would be better because it's much more naturalistic, but it makes it an order of magnitude more complicated. So I'll show you tomorrow. We have a new 3D tracking system in Constant, so we can do some of this, like sub-millimeter precision 3D. I think you're right. I mean, we have to. I mean, just ignoring one entire dimension of space is an approximation because of technological limitations. And it would be naive of me to say it doesn't matter. So the species we studied, by the way, are naturally found in very shallow areas, like 10, 15 centimeters of water. But they're also found in deeper water. We just always justify it by a natural part of their life is being shallow water, just because it's kind of hard to do the rest of it. Same with locusts in flight. I didn't tell you anything about locusts in flight because no one has ever studied locusts in flight. So I think it's a good point and it's well taken. And later on I will talk a little bit about 3-dimensional analysis of very small fish schools. And it turns out that they turn everything into 2D, which is, well, I was discussing it with someone yesterday. It's a bit weird, but potentially interesting. But you make really good points. Hopefully in 10 years' time, these studies will look trivial and that everything will be in 3D, hopefully. Okay, so let's go back in time. This is the 2005, 2011, because it does set the scene for some really recent work. Yep.
Okay, just have a question because you said in the fish network, this movement go to the better spots is what drives the movement, and it has this position that kind of how can I explain? Is the position that is like the most important thing in this network for other kinds of networks, like spread of information? Is there this kind of behavior that you want to go for the better spots? Like
I think they're always competing for the better spots, and the better spots are the front and the side edges.
I need to think more about my question.
Pardon me?
No, it's just because I was thinking for other networks, is there is this kind of idea of being in better spots in the network? For example, in information.
Oh, yes.
Better
I
if
think
you
I get
receive the information first or
right.
less for spread
Fantastic point. Sorry, I
news or things like this.
Yeah, I misunderstood you. So in our paper, when we study this, we actually compare it to Twitter and Facebook. So one of the obvious things is we don't want to have to invent a new metric for determining who's influential. Twitter and Facebook, and well, PageRank, for example, from Google as well. These are all algorithms, and I think there's one N something. It's too long ago since we did the paper, but we took the 3 most famous algorithms, like PageRank and the 2 that Twitter use, for identifying influential individuals, and applied it to the fish networks, and it does not work very well. And I think it's because these networks, you know, everyone is connected to, or the influential individuals are, like you have very few individuals who are super influential. It's like, you know, very few individuals are super wealthy. Like human society, modern human society, in terms of the structure of these networks, in terms of the distribution of wealth, it's completely bizarre compared to, you know, what we see within animal groups, where you typically don't get that type of behavior. So certainly, I would imagine our algorithm works better with, say, hunter-gatherer tribes or sort of natural communication, and the algorithms that have been developed for technology don't work very well for these natural groups because they're for these bizarre networks where people have hundreds or thousands or millions of connections, which simply couldn't exist in a spatially embedded network like this. So it's a good point, though, and in the paper, it's good you reminded me, because in the paper we did that because I think one of the referees asked exactly the same question, you know, and I think we gave a much more intelligent answer than I just gave in the paper. It's just too long ago for me to remember. No problem. Okay, so again, I'm showing you pictures of fish, but later on I'll show you these data with humans, with baboons, with all sorts of other creatures. But the general idea here was, you know, how can information flow within these systems? Now note, these studies were actually before the studies that I just showed you. So you kind of maybe bear in mind that, you know, I didn't know lots of the stuff I knew at the end of those studies when I did this, but I think this still serves a useful purpose. And so the idea here is that, you know, this individual may have performed a migration before and know to go in this direction, or it may detect a predator over here that the others don't detect, or it may have learned there's a food source over there the others don't know about. So there's lots of scenarios where there's differences of opinions within a group. Yet how does can this individual convey that to others? So I'm going to talk about leadership and collective decision making, first in animal groups, and then in the brain. So how do groups make informed, unanimous decisions? So does this individual need to signal to others, I know what's going on, follow me? Now in some systems there is explicit signaling, and we also study that. We've also got papers on that, but I took them out for simplicity's sake, because in many systems we have no evidence of explicit signaling. And I just want to show you that it's not required. Okay? You can also add in complexity. Also, individual recognition. Individuals need to recognize who is and who is not informed. Well, again, we have got very little evidence. These groups are constantly merging and splitting. The populations are in the millions, many millions. It's unlikely they know who is whom. So we wanted just to ask, how may they do this? So this individual has a goal-oriented vector. Now, at the time of this paper, I speculated that this was the case. What I'll show you probably this afternoon is direct neural evidence that this is true. And they also have social vectors, vectors pointing to the other individuals. Okay, so the model is deliberately simplistic, but it's a minimal model for a reason. And so the general idea is if these informed individuals, which I'm coloring in white, the uninformed are colored in green. Now, the colors are just so we can see who is whom. In the model, you don't know if anyone agrees with you or disagrees with you. And the big green cross just shows you the center of gravity, or centroid, center of the group. And in this example, these informed individuals have a common preference to move along the x-axis. And again, you can see with no signaling, with no individual recognition, they're pretty good at doing it. So we don't need to invoke that type of complexity necessarily. So if we have the same common preferred direction, but now we change the number of informed individuals within the group. So that was a relatively large number before. 1, 5, and 10. In fact, I showed you the data from 10. In a group of 100, you can see that one individual is not able to guide the group very effectively. 5 individuals, after an initial period of reorganization where they occupy the front of the group, they kind of guide the group in the right direction, but it's less accurate and less fast. And the 10 informed individuals I showed you before, spontaneously and super accurately, the information is conveyed. So we can plot this in a normalized fashion. From 0 is no information transfer, 1 would be perfect information transfer and going exactly in the right direction. But of course, this is a stochastic model. In all of the models, we add a noise term just representing neural noise, decision-making noise, and so on. And what we find, it's unfortunate that it's cut off. This is the proportion of informed individuals. So here's everybody's informed, 0 is nobody's informed. So this is the data from 1 individual informed. And you can see that for all group sizes, the accuracy increases asymptotically as you increase the proportion of informed individuals in the group. And if we look at, say, 85% accuracy, arbitrarily, you can see for a small group, like a group of 10, you need around half of the individuals to have information. But for a larger group, like a group of 200, we now need less than 5% of the individuals to have the information. We've simulated cell sheets. We've simulated migrating herds. There can be an infinitesimal proportion of individuals that know where to go, yet to the observer, it looks like everybody knows what's going on. And in tumors, for example, there are indeed these small number of leader cells, for example, in brain tumors, that lead the migrations of others. We also now know, and published a paper a couple of years ago on this, that most migrations, there's a relatively small proportion of individuals that are disproportionately influential. So we could also test this hypothesis. So these are real fish. The red ones are informed. The blue ones are uninformed. And what we did here was they don't respond to a green laser. For whatever reason, they haven't evolved with green lasers, I guess. But they can learn to associate a green laser with the reward. And so the informed individuals have learnt that this little green laser spot, which can appear with any of these feeders randomly, will be associated with a reward. And then when we do the experiment, we never present the reward. We just present the laser. And the blue individuals were uninformed. And you could see, actually, I know it's going through the whole thing again, but I want you to see the contagion, because we were just talking about social contagion. Watch the contagion. The red ones will tend to go to the front, and the contagion will spread through the group as a wave. And so we could see that as we increase the number of informed individuals, it increases, the accuracy increases very steeply, and then plateaus. Okay, that's cool. But in many groups, including human groups, there are differences of opinion, different motivations. Some are hungry, some are thirsty. Some want to go one way, some want to go another way. So can individuals in these groups vote? Can they make a decision together about where to go? Remember, they've got to our knowledge, no mathematical capabilities. They've just got these simple local rules. And so now we have a conflict between individuals within the group. And again, nobody knows if anyone agrees with them or anyone disagrees with them or how many opinions there are in the population. Can they come to a consensus? So I'm going to plot the data in the following way. Because what matters here are the number of individuals with different opinions, the strength of their opinions, but also the geometry of the problem. If the two individuals are differing in preference only by the two subsets, but only by a small amount, they can have it more or less in agreement. They could just average and go the right way. And so what we have here is one subset is always going to be fixed to go along 0 degrees, the x-axis. Then I'm going to change the other subset. If it's 0 degrees, it's the same model I showed you before. They're in agreement. It may be 1 degree, 2 degrees, 30 degrees, all the way up to 180 degrees, where they actually want to go in exactly opposite directions. And for every angle, for every degree, I run, I think, 40,000 simulations, and then show you where does the group go. The group will come to a consensus and go together. I'll come back to group splitting later on, but in a large region of parameter space, the group stays together and makes this decision. And so the exact numbers don't matter. Again, I'll show you later an analytic version of this model to show the numbers don't matter. But I think intuitively, this is just a group of 100. 5 want to go one way, 5 want to go another. So it's like the group below a critical difference of opinion, below a critical angle, here around 135 degrees, the group will spontaneously go in the average preferred direction, which is represented by the solid white line. So if they could do a perfect vectorial calculation, they would follow this solid white line. So they do a pretty good job until a critical point, at which suddenly they change from averaging the opinions to suddenly deciding between them. So if you imagine the groups actually moving through real physical space, there's a red target to the left, a green target to the right. As they approach the targets, the angle is slowly going to increase, right? And increase and increase. And once it reaches this critical angle, the system, the group, will randomly choose one direction or the other, right? Because there's an equal number with different preferences and there's an equal strength of preferences, and because it's a simulation. Now, have you discussed bifurcations in this course? Yeah, like dynamical systems or bifurcations? So this is reminiscent, at least, of a bifurcation diagram. And there are certain very interesting features that I'll get to later about what occurs near a bifurcation. The type of bifurcation this is, we have 3 different papers that propose 3 different bifurcations. A Hopf bifurcation, a monkey saddle bifurcation are the 2 most likely candidates, but it doesn't really matter for today's talk. We think it's probably a Hopf bifurcation. But here you're seeing the simulation, and it will randomly choose one target. In this case, it's red, but it would be equally white or red. Now, what do you know about bifurcations, right? This is going to be more intuitive to you than to a biologist, because these are random individuals. They're noisy, little noisy individuals that just have local rules. They start in random positions, random orientations. No one knows anything about anyone else. So you'd think just adding one extra individual with a preference to this group would just be washed out. But if you think about what we know about bifurcations, that might not be the case. So if we look at now 6 individuals going one way versus 5 going in the opposite direction, what we see is that below the critical bifurcation, the group will go in the average direction. But once it reaches that point, it's now no longer 50-50, but over 98% of the time, they choose the majority preferred target, even though no one knows what the majority is, and even though there's tons of noise, and even though no individuals can count. Right? So they're able to make extremely accurate majority consensus decisions.
They never divide?
I didn't say they never divide. I'll come
Sorry.
back to that later. This is the region of parameter space where they don't divide,
Okay. Sorry.
which is a very large region. So here there's one extra white individual, and now they will almost always choose the majority preferred target. And you can sort of see the local noisy nature of the interactions in the model. Yet the system itself is super sensitive. Now, what if their opinions are too strong? Well, then they can't make a decision. And in actual fact, what we now know is there's a sort of forgetting term. If you want to go that way, but you end up keeping on getting dragged this way, you begin to lose your preference that you had before. That turns out to be important, and it's in the paper, but it's not critical for today's lecture. So I'm going to focus on the times they stay together, because I then want to move on to the brain, and the brain, in general, stays together. We've also done studies where the naive ones can learn about targets and become informed, and there's lots of ways one can modify this. But when I wrote this paper, I wrote it again, you're so rushed by the editors, and it was a long time ago, 2005. And then we wrote it as, isn't it amazing that they can do this super accurate bifurcation, this super accurate decision making, despite all of those dumb individuals, despite all of those naive individuals. And it was only later when I was working on trying to understand my results on the evolution of these behaviors, I couldn't understand them. And I suddenly realized that the uninformed individuals actually play a critical role. I thought they were just going to be a source of noise. Let me just check that I have. And this, I don't know if any of you know Rand Paul, or have heard of Rand Paul. The libertarian is a very famous US senator. So he got very angry with me. I was on Fox News saying he was going to call Obama to cut all my funding for studying goldfish democracy. So, of course, he hadn't read the paper, he got the species wrong. I did not get $5.2 million to study this, I can tell you that right now. Yeah, it didn't go so well for him in the end. But he's a very famous US senator, also an asshole. So there's many, objectively, just look him up. There's many scenarios, right, where there are different opinions in a population. So I'm going to start off with fish schools, and then I'm going to move to opinion dynamics in humans, and then I'm going to move to opinion dynamics in the brain. So there are conflicting interests, and how can we resolve conflicting interests? So before, I showed you that if we have these informed subsets, that there's a competition between them. And you may have thought to yourself, well, what's to stop the minority from just becoming more strongly opinionated? And if they're integrating vectors, they could just have a stronger vector to point in a given direction. Now, firstly, this massively increases the chances of the group splitting. But even if they can make these decisions, let's have a look at what goes on. So let's have a look where everybody in the group has an opinion. Everybody has a preference. Now, in most scenarios in nature, that's probably not the case. But let's consider where there's 6 want to go one way and 5 that want to go the other way. And arbitrarily, so we have this term called omega. Omega is the strength of the preference, the strength, the length of that vector, basically. So if the majority preference is 0.3, then if the minority preference is also 0.3, then the majority will win. Okay, so you get this, what I showed you before, the majority tends to win. But if the minority become more and more strongly opinionated, they can begin to, here is 50-50, half the time the minority wins, half the time the majority wins. But you can see if the opinionated individuals become so strongly opinionated, they can get their own way at the cost of much more splitting. So it's much harder for the group to come to consensus, but if they do come to consensus, the minority, so being opinionated seems to work. But remember, this is if everybody has a preference. What about if you have individuals in a group that have weak preferences or no preferences? They could be perfectly well informed. They could know about both options, but not care which one is the outcome. Or they may not know what's going on. Okay? And so previously, we and others have thought that they're just going to add a source of noise. But that's where I love doing science, because you can get all of these surprises, because that was not true. So here, if the minority have a stronger preference than the majority, 0.38 to 0.3, and there are no uninformed individuals, the minority will, the proportion of groups reaching the majority will still tend to win here. Okay, the minority doesn't have a strong enough opinion strength to win. But note what happens if I add 10 or 12 stupid individuals or uninformed individuals to the group. It pushes further control to the majority before, if there's too many uninformed individuals, the group can no longer make a reasonable decision. But what's more interesting is if I increase the minority preference further and further still, now the minority is so strongly opinionated that in the few times they can reach a consensus, because most of the time they split, the minority will win 98% of the time. But if I put in, again, this small proportion of around 10 or 15% of uninformed individuals, not only does it act as a social glue and stop these fractions from never seeing eye to eye, it helps them make decisions, but it spontaneously returns control to the numerical majority. That is, the strength of opinions is negated, even though it's not changed. So it's an emergent property. So we thought, this is really cool, because this could prevent an arms race of opinionated behaviors within natural populations. I'm not talking about human populations here. But we did want to know, is this a general principle? Because there are many systems where individuals influence and are influenced by others. There are technological systems, there are algorithms that influence each other. There are humans that influence each other on technological systems. There are neural systems, and of course there are social systems. So in our paper, inspired by this, we then decided to do different models that have been used in the social sciences, one of which I'll present here. And it's a very, very simple model. Again, always use the simplest model to see if the phenomena is robust. And so this is just representing two individuals on a network. Now this is now non-spatial. The network interactions just represent who's interacting with whom. And so these two individuals interact. One has a red preference, one has a green preference. Of course, you can think of this in any type of opinion space. And they each have a small probability of changing each other's opinions. But of course, individuals don't just interact with one other individual. There's the complex contagion idea. What about if you interact with more than one? All we assume, and that's all that's needed mathematically, and by the way, this can work just with 2 and 3 interactions. In the paper, we look at much larger numbers, but all of the maths can be done with 2 and 3. And now the red one, if there's a probability of being convinced by green by Q for one individual, now the probability is slightly greater than 2 times Q. There's a synergy here. That's the only mathematical thing you need to put into this model. And it increases probabilistically the probability it changes. It's not deterministic, it's probabilistic. So it's not so interesting with 3 individuals. What if we have 10,000 individuals on a network interacting in this way? And I did this work with Thilo Gross and Govin Demirel, and I kept them blind to my predictions. And so when they sent me this graph, they were really confused. I was really, really happy, because they showed that if the majority and minority have an equal strength of opinion, the majority wins. If the minority become increasingly opinionated, either unwilling to give up on their views, or if they do flip their views, they're more likely to flip back, both give you the same results, they can dominate, as we saw before, the decision making. The minority can win. Now, if we look at the percentage of uninformed individuals on the network, initially nothing happens. And then at a critical point, a tiny proportion of individuals that don't care what the outcome is completely dictate what the outcome is. And the system flips to a majority decision. And because it's a large system size here, it's a very, very strong nonlinearity. Now, working with Thilo and Govin, they used moment expansion and moment closure to actually prove this result. So this is well beyond my capabilities. But the point I want to make is that the simulation and the analytic model were very much in agreement with each other. The simulations are the circles, the analytic model are the solid and the dotted lines. And so you can now see this bifurcation, where there's the stable minority, and then there's the appearance of a saddle node bifurcation and an unstable solution here, and a stable majority solution there. This is the unstable, undecided state. And so the nice thing about this very beautiful mathematical model is it sort of strips away all the complexity. There's no space, there's just these sort of simple network properties. And then we can look at what's really important, which is the relative strength of the minority preference, how much stronger their opinion is than the majority, and the numerical advantage of the majority. Those are the two fighting forces here. And you can see that with no uninformed individuals, the minority can dominate if they're strongly opinionated enough with respect to the numerical majority. But now what I'm going to show you is the region of phase space that flips from minority control to majority control. And I'll show it in white. So now we're adding the small proportion of uninformed individuals, or unbiased individuals. Now the majority wins everywhere except this little thin sliver. The minority now really struggles to win. So being opinionated can work if there's no diversity of opinions. But if you have diversity of opinions, especially if individuals with weak opinions or who are not, it's hard to sort of express it, is it's not that they're uninformed, or they're unbiased. It could be just unbiased. They could be well informed about both options, but actually listen to both sides of the story, so to speak. And all too often, I think, some individuals tend not to be. They tend to be very polarized. And so we found this in a whole series of papers. We've since studied this in coupled oscillators, in spin systems. Yeah.
Of transition, what happens is there are, it is equally likely for the result to be what the majority wants or what the minority.
I can't quite hear you. Sorry.
It's not?
Yes, it is. It is on. Yeah, it's just it's much clearer now. Thank you.
In the region of transition,
yeah.
What happens?
Like at the phase transition.
Yes,
you mean?
But then afterwards you showed us a white region which was very large.
Yeah.
In that region, what happens is that
the majority always wins.
Oh, also in.
So this is. So I could have colored it grey.
Okay.
Yeah. So you can think about it as being grey because now the majority wins here and here. So this could all be grey, for example. Okay. I just colored it differently to show that this one flipped to this state. So the majority wins everywhere in phase space, and the minority only wins here.
Okay.
So basically, it's prevented the minority strategy from winning.
Okay.
Yeah. And so we've shown in a series of models that a small proportion of unbiased individuals increase the probability of reaching consensus because they act like the social glue that prevents the groups from, like in the physical groups from splitting, but in the sort of social groups, ideological splitting, where you just will never see eye to eye. They act as a means of communication between more extremist subsets. They increase the speed at which consensus is made. They make decisions much faster. And the sensitivity. They make, for smaller differences in preference, they can come to a consensus more accurately. But of course, I'm an experimentalist, and I'll come back to why I think this theory is so cool. But it also makes a testable prediction, because if we then go away back to the flocking schooling model, you know, we now are kind of trusting our model, right? We see this in all different types of mathematical frameworks. But if we go back to our schooling model, we have a prediction that adding uninformed or unbiased individuals should inhibit the influence of a strongly opinionated minority and return control to the numerical majority. And so, as I said, doing experiments is always really hard. So I'm going to show you 3 data points that took us about, oh God, more than 9 months. It took Christos Banu, who's now professor at Bristol University. He was a postdoc in my lab when we did this. And so Christos ran these experiments where we had fish trained to a blue target or a yellow target. And yellow is like crack to a fish, right? They just train really strongly to that target. So that's going to be our strongly preferred color. And then we have our weakly preferred color. And then we can mix combinations of individuals. And even though we're now talking about really small system sizes, we're talking really small system sizes, you can still see the theory predicts a strong nonlinearity. It's not this complete step function we saw before, but a very strong nonlinearity from the theory that the minority, if they're strongly opinionated, control the group dynamics, but then adding 5 or 10 uninformed individuals should flip the group, which is very counterintuitive. No one would have thought to do this type of experiment. So here's the three data points that Christos worked so hard to get, because it's hundreds of experiments. But here you can see that if the minority and majority have no uninformed individuals, the minority with that strong preference will dictate what the group does. But then if we add 5 or 10 informed individuals, exactly as we predicted, it flipped the dynamics. Now, importantly, Christos was blind to the predictions again, because you never want an experimentalist to know what you're hoping to happen, because he might want to please me, or then sort of see the results. So admittedly, we film all of it, we get as much quantitative data, but there can always be intrinsic biases. So when he came to me, he was also terribly disappointed with the data, and I was the only one happy. So really, this is going to be something that comes back again later. Diversity of opinion strengths promotes democratic consensus decision-making. We often exclude individuals without strong preferences from decisions. Note that the uninformed individuals are part of the decision-making process here.
In the last experiment, the majority was blue. You preferred the blue, and the minority preferred the yellow. Was it?
Yeah.
Okay.
I mean, it doesn't really matter.
Yeah, because the yellow is like correct for the minority, but as you mentioned.
Well, at the time, this is what we thought. The yellow, they train very strongly to yellow, but we don't think the colors are necessary for the experiment.
Oh yeah, sure. And the blue preferred not so strong preference, but still.
Yeah, they still have a preference. Yes, exactly. In fact, we were probably wrong that it was the colors. So the colors definitely, right? They learn much more quickly to yellow than they do to blue. But because we trained them for so long, they actually didn't care about the colors. You could run this experiment with no colors at all. They actually preferred different locations in space, which is kind of interesting. But you could do this in lots of different ways. I mean, in principle, all you need is a stronger preference. So you could, for example, have hungrier individuals and so on. We just thought of the simplest way of doing it. But any further questions? I think that's us out of time for this morning. But if there's any further questions, otherwise we'll come back to how this can teach us about the brain in a moment, in the next lecture. Yeah? Cool.